everyone, this is Hannah from MyStem Kits, and I'm going to talk to you today about our DNA kit. This is one of my favorite kits and I'm really excited to tell you about it. So let's talk about assembling it. First, you're going to need to take your sugar phosphate backbone pieces here and desupport them. Simply use your fingers and break off the supports. Once you've done that, look at your nucleotide bases. Now these have indications on the back that indicate whether or not it's a purine or pyrimidine, and on the front they have a letter that indicates which one it is. So guanine, cytosine, adenine, thymine, and then when you're doing RNA, uracil. And so the kit comes with 18 of each base pair, which gives you a few extras depending on what you want to do. That way, in case one of your ball joints snaps when you're connecting it or something, it's not going to be an issue. You'll just have replacements built right into the kit. So, to assemble it, line up the pieces you're going to want to use in your DNA chain, including 3' and 5'. Once you have them, you can simply snap the nucleotide bases onto the sugar phosphate backbones. Now, if you have something that's a little bit loose and doesn't give you that good snap connection, you can just use a little bit of tape and wrap it around the circle on the sugar base, and then when you snap it in, you get a tighter connection. Once you have all of those, you can snap the sugar backbone pieces together to form a chain. Don't forget to add your 3' and 5' to the end to indicate directionality. Once you have one chain, mirror it using a nucleotide chain on the other side that has the matching nucleotide bases. Once it's all connected, snap them together to form a chain. One thing to note when assembling it is that the ball joint between the cytosine and the guanine is different than the ball joint between the adenine and thymine. Just like in the real world where adenine cannot bond with cytosine, in the same way the ball joints here are two different sizes that they will not connect or bond together. Once you have your complete chain built, you can just see how when you double helix it, it can condense and take up less space. Or you can talk more about how the processes of DNA replication, transcription, and translation work, and how even a simple genetic mutation that happens during replication can carry on all the way through transcription and translation into protein synthesis to give you the wrong protein, which can have side effects on the human body, for instance with sickle cell. As you can see, all the different things you can do with this, going in, doing DNA replication, then once you have your duplicated chain, you can go in and do DNA transcription where you're then forming a messenger RNA strand. That's where the uracils come in. And then take that messenger RNA, tie in your anticodons with your tRNA and actually connect them in, build your protein chain, and see how you create this protein macromolecule of amino acids you saw from development all the way from DNA replication. So whatever you're going to do with it, whether you're using it in middle school, high school, or beyond, it's a really great kit to have in your classroom. Thanks for listening.